Good morning, everyone. It's a pleasure to be here, and a big thank you to David for organizing this really interesting meeting. I've been talking to David for a while over Zoom, so this is my first opportunity to actually meet him in the flesh. I also have a sort of other debt that I should mention. As Ravi said, I'm a mathematician by upbringing. I'm actually a pure mathematician. I identify as a pure mathematician, I should say. My lab has been studying essentially cellular information processing over a number of years. It's only rather recently that I've become interested in this question of cell learning, and this has been partly a collaboration with my colleague at Harvard, Sam Gershman. Some of you may know him. And Sam and I have been organizing a discussion group on cell learning that meets remotely, has been meeting remotely over a number of last couple of years. David and also Randy Gallistel have been sort of very important members of this group, so I owe them thanks for helping me sort of navigate the terrain between systems biology and cognitive science. That's been... David, I just looked on the web. You look much more distinguished now, I have to say. And I'm really very glad to meet in person several of the people who've actually participated in this discussion group over time. So if anyone's interested in joining us, we're always on the lookout for new recruits, so please drop me or Sam an email. So one of the things to come out of the collaboration with Sam is this perspective, which actually Hessam mentioned in his talk, which puts out the case for thinking about learning at the cellular level, and particularly tries to bring out the work of Beatrice Gelber that Hessam mentioned. But I'd like to draw your attention to another paper a perspective that I wrote recently, which is published in a place where no biologist is likely to find it. This forms the backdrop for some of the things that I'm going to talk about. It's discussing learning on a broader scale, but I want to focus today on the question of what happens at the level of individual cells. So let me begin with a little bit of personal sort of history, how I sort of got started in this. It goes back to work that the American biologist Herbert Spencer Jennings did in the very early part of the 20th century, around 1900. And Jennings described the behavior of a single-celled protozoan, a stentor roselli. So in Wallace Marshall's talk yesterday, you would have seen a cousin of stentor roselli. This is stentor ceruleus. And there's a very important distinction between these two organisms, by the way, although they're very closely related phylogenetically. Roselli, it's this trumpet-shaped single cell. It uses cilia to create a vortex to draw bacterial particles into what's essentially a mouth. It's typically sessile. It anchors to a holdfast, and it likes to remain there. And what Jennings found was that if you irritate stentor roselli, he did it with squirting carmine dye at them through a pipette. They go through an extraordinary series of avoidance reactions, which are depicted in the picture there. They first bend away from the stream. And if that doesn't work after a little while, they alter the direction of rotation of their cilia. So instead of the vortex, the hydrodynamic vortex, bringing particles to the mouth, it expels particles from the mouth. And they do that, and if that doesn't work, then they perform an extremely sharp contraction, the same contraction similar to the one that Wallace was looking at in his studies of habituation. They contract onto their holdfast. After a while, they reemerge. And if you keep irritating them, then it's very hard not to use anthropomorphic language here. They basically get fed up, pull up their holdfast, and swim off. So all this is happening in a single cell. And I was entranced when I first came across this, actually, a lecture of Dennis Brain, which he mentioned it. And I was really horrified to discover that in the community of people who study learning, Jennings's experiments were thought to be, were believed to not be reproducible. And in fact, it was someone who Wallace mentioned, David Wood, who is in fact a very fine experimentalist who did a lot of work on Stentor, who told me that Jennings's work was not reproducible. I was really disappointed. And something didn't seem right about this because I'd read Jennings's papers, and he's a beautiful biologist. He's, you know, a wonderful experimentalist. And I kept thinking, how did he get it so wrong? He worked on these organisms for years. He did these experiments repeatedly. How could he possibly have not got it right? And eventually it rankled so much that I actually persuaded myself to go into the depths of the Countway library, dig the paper out of the stacks. It was a kind of Dickensian experience, and photocopy it. And I discovered it to be the single worst experimental paper I have ever read in my life. And I invite you to read it yourself and come to your own conclusions about it. So to begin with, they didn't even use the right organism. They couldn't find Roselli, so they thought they would use the Stentor ceruleus that Wallace was talking about. Now, Stentor ceruleus does anchor to a holdfast, but it's typically motile. If you irritate it, as Wallace told us, it swims away. So it's hardly surprising that they weren't able to reproduce this behavior in ceruleus. Now, I don't want to get into the details of what we did. I mean, basically, this started off a skunkworks project in the lab, and eventually we did the experiments and confirmed that Jennings is right. There's all sorts of, you know, kind of details. You can read the paper. This avoidance hierarchy is real. You can go and watch the videos and you can see it for yourself. Okay, so the thing I wanted to sort of mention is, why is it that people decided that Jennings's work was not reproducible, right? And having read the literature in some detail, I can only come to the conclusion that it was for ideological reasons. Basically, the community of people who were working at the time, basically in the 70s, didn't believe that single cells were capable of complex behaviors. And they preferred to believe a really shoddy paper, because it said the right thing, to a long series of really good papers that said the right thing, the wrong thing, right? I'm not a historian or a philosopher, but I regard this as the only explanation I can come up with is that ideology trumped evidence. And, you know, I'm a mathematician, so that means I'm pretty naive, particularly about biology, but this was really a shock. It's not supposed to work this way, right? But I think perhaps it does, and I think this touches on some of the things that David talked about in his introduction, and also David in his talk. But I think I would point out here that I don't think there's a psychological explanation for this. I don't think Jennings was a sensationalist. He was actually a very conservative, sort of mainstream biologist. I think the reason for this is sociological, not psychological. And I don't think we like, as a community, to believe that we are prone to sociological pathologies of this nature. I fear, however, that that is the case. Now, I don't want to sort of dwell on this. I'd like to just say that I drew two lessons from this. The first is that if we're going to come and explore these issues of learning, then there is an obligation on us, first of all, to be very clear and precise and rigorous in the definitions that we use. And not only that, but we also have to have an experimental program or a direction that others can follow to develop and confirm the work that we do. So those two requirements I don't think are sufficient to avoid the pathologies that I'm pointing to here, but I do think they're necessary. And that's really guided what I want to sort of talk about today. Okay, so in that spirit, what do we mean by learning? Okay, so if you open the psychology or cognitive science textbooks, you'll often see a sort of informal definition that says learning is a change of behavior in response to the same stimulus, and that's actually, you know, kind of not a bad, quick summary of what we think of as learning. But typically what people will point to are the classical protocols for thinking about learning that go back to behaviorist psychology, non-associative learning, habituation and sensitization, and associative learning, the different forms of conditioning that you're, I'm sure, all familiar with. Now, these are very important, and as we saw in some of the talks yesterday, we still rely on these experimental protocols to study learning. We saw that in Wallace's talk on habituation. We saw that Colleen mentioned her work on learning, on conditioning. So it's not to say that these are not practically very important for us. However, I do think they're completely inadequate as a way to think about learning. And I think the first thing that we have to do is to extricate learning from the embrace of these particular experimental protocols and the embrace of sort of behaviorist thinking, and to consider it as a form of information processing. Okay, so I think that is the first starting point. So what I'd like to suggest is a definition that we might be able to use in order to sort of define learning in a rigorous way. Okay, so so this is very much a work in progress, and I think it clearly draws on things that other people have suggested. I'm sort of putting the pieces together in a particular way which I think is useful. And there are sort of two main parts to this definition that I'd suggest. And the first is kind of obvious. An entity that's learning has to acquire information from its environment, and we can measure that very accurately and rigorously in terms of mutual information. And if you've forgotten what the formula is for that, I've written it down so that we have it. So this is something that we can get our hands on fairly clearly. But there's a second part to this, acquisition of information. And this information that's acquired needs to be represented in some way within the entity. And my preferred language for talking about this is that of an internal model. And I use that because I think that's, in fact, actually a very evocative way of thinking about what this representation is. Now, this internal model is a form of memory in the sense that it typically can carry information forward in time beyond the persistence time of the information in the environment. Okay? So in this way of thinking, learning and memory are very intimately related. If you learn, you have to remember something. If you remember something, you must have learnt it. Okay? Now, finally, and I think this is very important, this last part, this representation, this internal model, has to be causally responsible for some behavior in the organism. And I think this is the critical thing that links it to what was said on the previous slide. What does it mean to be causally responsible for something? It means that if the information was not present, the organism's behavior is different. There's a change. And that is the issue of change is what links this to what was said on the previous slide. However, I think it's more general, because it allows for the possibility that the change that is observed may result it may come about because of a different stimulus to the one that was originally received, or in a different context for the organism. Maybe an internal context is different. Or it may be cryptic in the sense that the context that elicits it, or the stimulus that elicits it, hasn't actually been received. So if you think about being infected by some bug, your immune system becomes primed, but you don't notice what the immune system is capable of doing until you get a second infection, right? And if that never arises, I don't think it's sensible to say that the immune system hasn't learned something as a result. So this definition, the words are carefully chosen to allow for this, but I would reiterate that this is very much a work in progress, and I welcome suggestions for how this can be achieved, and it's always very fun to think about sort of corner cases for these kinds of definitions. Okay, so that I would like to propose is a definition of learning that's both rigorous and something that we can experimentally take forward, and I think that's very important. Okay, so I want to make a few asides here. I'm going to ignore several very interesting aspects here, but I don't want you to imagine that they're not important. When we talk about internal models, I think we have to remember that the cell itself has a lot of internal state, and it's inconceivable that the internal models don't rely on that internal state. We mostly don't sort of worry about this, but I think it's terribly important if you can think a bit, if you like, in a Bayesian perspective as a kind of prior for how information is being interpreted. Okay, so a second very important thing is that it's all very well to think about the internal model representing information, but information in the environment is always changing. So how is the internal model being updated? Now this touches on issues that were raised yesterday in Wallace's talk about forgetting and memory, right? I think in the neuroscience literature, there are some very interesting sort of normative ways of thinking about how memory is updated. Carl Friston talked about this in the context of predicted coding and free energy minimization. I find this a very attractive idea, the idea that internal models are updated to minimize their prediction errors. I think that would be wonderful if we could show that, but I think the first thing we have to do is to find the internal models in the first place before we go down this road. So I'm not going to sort of pursue this very interesting issue of updating. Now, finally, I think if we do have internal models, a very natural question is what kinds of computations or information processing are done on those internal models. And to me, that offers a way to approach the question of cognitive capabilities. So in some sense, this is the other way round to, I think, this idea of basal cognition, which Pam and others have been thinking about. I see learning as being a more fundamental way to approach cognition rather than cognition as a way to approach learning. And I think that because learning has this, we can give a precise definition to it and we can experimentally explore it. Right? So I'm not going to sort of pursue these directions, but I wanted to sort of mention them here. Okay, so that's all very well, but are there any internal models? Well, what I'd like to tell you, this is the next thing I want to tell you, is that they're hiding in plain sight. In fact, all of you know an example of learning at the level of the individual cell, which clearly obeys all of the conditions that I laid out in the definition. It's not called learning. I've played this game a couple of times, and usually nobody guesses it. So I won't play the game again. But you all do know it, right? It's called CRISPR. It's not the cut and paste technology for DNA. It's the innate immune system in bacteria. And you all know how this works. A single bacterium is infected by a phage virus, and the DNA from that virus is cut and pasted into the bacteria's own DNA. That's the CRISPR locus. And if there's a subsequent reinfection, that locus is used to generate an RNA that then destroys the invader. So all of the conditions of the learning definition I gave are satisfied by this example. And I think this is a nice example because it makes it clear how something that we know very well looks very different when you look at it from this other perspective, this perspective of learning. And we never think of it in those terms, although the immunologists sometimes use words that are borrowed from the psychology literature. It's basically just words rather than actually using it in a scientific way. However, I think this example is a little bit misleading in some ways because in this case, the internal model is DNA. And I think that's actually the wrong place to be looking for internal models. I think we've been sort of spoilt by the fact that we have such enormous understanding of polynucleotide as forms of memory. We understand very well how hereditary information is encoded in DNA. And notwithstanding Hassam's beautiful discussion of universal computation on polynucleotides, I actually think the kinds of internal models that we're looking for are very different in character. They are spatiotemporal, they are distributed, they're dynamic, they're transient, they're stochastic. And we have to think about that in the context of all the complexity, the molecular complexity that's happening within complex cellular environments. And once again, I think these models are hiding in plain sight. The current literature has examples of these. And I'm not going to get into this in detail. These are just some of the papers where I think if you read them carefully, then what you see is that people are uncovering exactly the kind of internal models that I'm alluding to in this definition of learning. We've already seen one of these in Aneta's talk. That's the paper with the blue font that was on the slide. Now, what's very interesting is that you'll notice that the titles of these papers include words like memory, information, and encoding. None of the papers use the word learning, except one, and that's the one at the bottom from Elaine Fuchs. It just touches on the word learn. Now, I find that very curious, because as I was saying, in the way I'm thinking about it, memory and learning are two sides of the same coin. The papers are all very different. They're very different contexts, biologically speaking, and they don't refer to each other. But I think when you take this learning perspective, we can see that they're all essentially trying to do the same thing. It's just in very different contexts. Now, I'll give you a quick look at one of them. And this is a very nice paper from Peter Swain and Gasper Taksik. It's a study of how information is encoded by yeast cells during different forms of stress response. And really, the picture there says it all. They look at a panel of 10 transcription factors in yeast, all of whom are implicated in the yeast stress response. And you can read for yourself the very actually crisp and clear sentence that they write there that summarizes their findings. And you will see that it basically encapsulates very much what we've been talking about, that the spatiotemporal, what's been plotted here is the nuclear cytoplasmic ratio of these transcription factors, so where they are, whether they are in the nucleus or in the cytoplasm over time. And if you have that information for these 10 transcription factors, you can basically tell what external information was present in the environment. That's basically the encoding. Now, they don't look at the question of the timescale of the memory. That would be relatively straightforward to do. They just didn't think about doing it. And the question of causality, they didn't look at directly. They didn't sort of modify the information encoding and then look to see what the downstream effect would be. But all of these transcription factors are well known to be implicated in the stress response, so it's not unlikely or implausible that it would have some kind of effect like that. But they didn't search for it. Okay. So this, I think, gives us an idea of how very different the kinds of internal models are that we might need to look for. It's very far from the nice sequential information in DNA, right? It's distributed, dynamic, transient, all of those elusive things that we have to get our hands on. Okay, so very quick. So this is really my way of saying that these internal models are there. We just have to try and find them. What I do think that these papers help us understand is the kind of experimental methodology that we might use to get them. And this is a kind of summary that extricates from the literature that's present a kind of strategy for how we might get our hands on these internal models. I think there are a couple of important points here. One is the first one, which is that we need very rich environments. The typical kinds of studies that we do in signal transduction, molecular biology, we have a dish of cells. We basically put a single bit of information in the environment. We give them a saturating concentration of some ligand. You know, if you're trying to correlate that with information in the cell, that's hopeless. So I won't go through what you can read there. It's fairly straightforward. I just think that what we're aiming for here is to end up with the equivalent of a coding table for these internal models. And you all know the coding table for protein coding genes, and I think what we need is sort of the equivalent that says what information is being represented and what causal impact does that information have on the organism. And I think this is a strategy that we could, in principle, execute now. So very quickly, what's the point of doing all this? So I think there are several things that one can say on this. I think, to begin with, that understanding how individual cells learn is a foundation for understanding how learning is operating at other scales in the organism. And we've talked about neuroscience and the problem of the engram, and we still don't know what it is. And absolutely, I believe that without understanding what happens at individual cells, we're not going to work this out. Immunology is another place where we know that learning is taking place. Mostly we think about clonal populations, but there's also increasing understanding that we have to see this at the individual cell level. And there's a very interesting evolutionary direction here, which I won't sort of bore you with. I do think that this idea of internal models and the computations on them allow us to potentially think about what's happening in cells at an algorithmic level of description. And if you're familiar with David Marr's 3 levels of explanation, I'll leave it at that. I do think there are sort of very broader general things that we can say about why learning is important, and I'll leave them on the board just to tease you with. But let me finish with a final point, which is that I think we've been living in biology with an extremely successful metaphor. It is the metaphor of the selfish gene, which goes back to Richard Dawkins' famous book. This is a brilliant book. It's beautifully written. This is, I think, an outstandingly successful metaphor. I think it's high time we got rid of it. Okay, I think the very sharpness of the light that it has thrown on biology has thrown into shadow all the things that don't fit into this, and that we need a new metaphor that can compete with this. And I'd like to suggest that the learning cell offers us a much more productive way to think about biology in the future. I think thinking of cells as entities that can construct internal models and minimize their prediction errors, I think is a much richer perspective to use in thinking about how biology should be in the future. So I'll leave you with people in my lab, all of whom are a little bit sort of scandalized by this new direction that we're taking. But the people who've been persuaded to contribute to it are highlighted there. So thank you very much for your attention.
Thank you. We have around 3 minutes for questions.
You go.
Okay. Nobody else? I always ask questions, so I want to let everybody go. So Jeremy, that was fantastic. I was just curious, I was going to push on your definition a little bit, because I'm wondering if the notion that you have to have an increase in the mutual information is precisely appropriate. So in the case of Colleen's talk yesterday, we have worms that sort of might have the same mutual information about their environment, meaning that if you look at their decision index or whatever, and you calculate the mutual information there, they originally prefer Pseudomonas to whatever, but then they change that. So the mutual information hasn't increased, but something about the sort of probabilistic structure of the thing has changed. Does that make sense, what I'm saying?
So you'd be happier with change rather than increase?
I don't know. Or maybe the notion might be to actually focus on that objective function thing, that you're, the error, that thing as being, but that has problems too, so I'm not sure. But it just a change in the probabilistic, like a change in the types of decisions that are being made or the types of behaviors that are being observed, maybe that's slightly more appropriate than an increase in mutual information. I'm just throwing it out there.
Oh, no, I take your point, Eric. And I think what the definition was really trying to get to was that information needs to be coming from the outside to the inside. And if that might happen through a change in the mutual information rather than an increase in it, then that might still be a possible way to do it. So I'd be happy with that.
Hi, I'm Emre Alka from the University of Toronto. You mentioned something that I'm very sympathetic to, but I want to sort of ask about having it expanded on a little bit, which is speaking about to understand the causal relationships in learning, we need to give our specimens a rich environment. And I'm very curious about if you could expand on a little bit more of what you mean by a rich environment, and then if we give them one, how would we actually make causal descriptions?
I actually wasn't saying that. I do think that we need the rich environments in order to be able to understand what's correlated inside the cell with what's outside. If we put a single bit of information outside, there's absolutely no hope for understanding what it's correlated with, because we correlate it with everything. So without rich information in the environment, we won't have the ability to identify what inside the cell is representing the information. That's the first point. Now, the causal issue, I think, is a very different one, and I think requires very different experimental tactics in order to get our hands on. And there, I think it really comes back to doing, sort of very conventional molecular biology and genetics, which has, have very powerful tools for understanding the causality among molecular players. Yeah, so I think those are sort of two different aspects of the strategy, but one needs both if one's going to get a handle on the internal models. Yeah.
One last question there. One last question. We have a minute.
Jeremy, thank you so much. That was really exciting, and I've got so many questions, but principally, transcription factors. You say they can encode enough information to unambiguously report on environmental change. How do you think it gets there?
How do I think
the information gets into the transcription factor?
Yeah, I mean, that wasn't specifically explored in the paper that I...
Not at all.
Yeah.
I mean,
But-
but there's been a lot of talk about epigenetics here, and a colleague of mine whom I think you know is working on something in relation to the Baldwin effect and Stentor Roselli's complex avoidance sequence.
Mm-hmm.
And I was wondering if you had any observations on that, or is that too far off-piste?
No, not at all. I mean, I think it's plausible that the information that is acquired from the environment during processes that we might call learning are not in one place, but are actually being transduced between different... there are different internal models that are operating at different timescales within the cell. So at a very coarse level, one might imagine that the information is first present on receptors, and that the information that is present is encoded somewhere very close to the receptors, perhaps in signaling pathways. Eventually, that information impinges on the genome, and then perhaps is re-represented in the form of transcription factors. And that, I think, is what the Swain paper was observing. It is that time point during the process of information transduction. And it may be that there is a memory that's much longer term, and we might imagine that that was being encoded perhaps by epigenetic mechanisms. So I think just as we do in the cognitive world, in psychology, we imagine that learning and memory operate on very different timescales, we should think of the same hierarchy within the cell.
Okay, let's thank our speaker again.